Hi class! In this video lecture we're going to look at sections 13.1 and 13.2. So 13.1 looks at really the carbohydrates again. We've looked at simple sugars already. Now we're going to take a deeper look at the polysaccharides and the glycoconjugates. So looking at, for example, the polysaccharides and glycoconjugates, what are these and how are they different than what we've already looked at? Well, if we start classifying and grouping our carbohydrates, the monosaccharides are going to include things that we've already talked about: glucose, fructose, galactose, mannose, etc. Whereas the disaccharides that we looked at, being sucrose, maltose, lactose, we start combining those individual monosaccharides into two pieces with that glycosidic bond. The oligosaccharides are going to include short carbohydrate pieces up to about 10 individual monosaccharide units, and then we start classifying them as polysaccharides or these glycoconjugates. So the polysaccharides can be further classified into what we call the homopolymers versus the heteropolymers. The homopolymers are going to have the same sugar unit or monosaccharide derivative repeating over and over and over. Whereas the heteropolysaccharides are going to have two or more, so 2 plus, individual monosaccharide units that repeat. These disaccharide components can include other modifications, as well as the homopolymers can have some modifications of what we've already seen. So we're going to look at some other types of modifications on our polysaccharides, but again, the homopolymers, same repeating unit, the hetero, 2 or more different monosaccharide individual units that are going to be repeating. Polysaccharides are different than the way that we think about most molecules in chemistry. They don't have a molar mass or a molecular weight. And you're like, wait, what? These are so large that, and they really don't have any defined ending, that they do not have a molar mass because of that. So keep in mind, these are going to be extremely large molecules. The glycoconjugates are going to include pieces that are saccharide derivatives with a piece that is not. So glycoproteins and proteoglycans both have amino acid portions as well as the monosaccharide or the saccharide polysaccharide portion. How do we distinguish between these? Well, it gets a little confusing. But if you put an emphasis on the last part of that definition, that's going to tell you what the portion of greater mass or greater amount is. So for example, the glycoproteins, these are going to have a small carbohydrate portion and a large, most of it is going to be made of amino acids, whereas the larger portion is the protein. A proteoglycan, on the other hand, is going to be largely composed of carbohydrates, 85%, 95% as an example, and have a smaller amino acid portion that might be a peptide or a very small protein. Glycolipids are going to be some sort of sugar or carbohydrate moiety attached to a lipid. And we haven't looked at lipids yet, but we're getting there fairly quickly. And you can see some of the examples that they give and the importance of these. The glycoproteins included in antibodies, viral coating of viral proteins. Proteoglycans are going to have a variety of different roles that they play. Glycolipids oftentimes are going to be bound into the membrane of a cell. So shown, for example, right here, where we have a glycolipid, the lipid is going to be membrane bound and anchored, and then the carbohydrate portion coming off that lipid into the extracellular matrix, as an example. So just starting to take a look at the glycoconjugates. The glycoconjugates are going to include things like glucose and galactose, some of these sugars that we've already looked at. Let me make that a little bit bigger so it's easier for you to see. There we go. But you'll notice that what is done here is we've made kind of a code. So the consortium for the glycobiology has put together this coding system where we can quickly show these different saccharide units using a colored symbol. So glucose being the blue circle, galactose the yellow circle. Instead of having to draw out the full molecular structure for each one of these different saccharide units, now we can start linking these together and making a code of it. You'll notice that I've starred the ones that are the most important. The N-acetyl linkages onto glucose or galactose are going to be shown in the same color, but now we've made them a square instead of a circle. So an N-acetyl glucosamine or N-acetyl galactosamine is going to contain, through the nitrogen, a link directly to that carbohydrate monosaccharide. So it's linked through the nitrogen, and then the nitrogen linked to the acetyl carbon. Quick note here, these are technically classified as amino sugar derivatives, but the actual functional group is going to be that amide functional group. So keep that in mind. Other derivatives that you'll see, carboxylic acid sugars will have, so I have an arrow pointing to that carboxylic acid on the N-acetylneurominic acid, where we have that oxidized first carbon as a carboxylic acid. So it's decorated slightly differently than we'll see some of the others. The glucuronic acid above it has the same carboxylic acid derivative of a sugar. So we can decorate our carbohydrates in these different monosaccharide units with these different functional groups, and that's going to obviously change their reactivity, changes their chemistry, changes their intermolecular interactions between them. So I'm just going to label these carboxylic acid sugars as well. And what that results in is a sugar code, if you will, for cells and proteins that allows them to be distinguished from each other. We'll have receptors for some of these very specific modifications, but it's also made this field of research somewhat not only interesting and important, but also complicated in that we can change one functional group on these residues that are already so structurally similar and already so very similar based on their mass sizes, if you will, of an individual unit. You send them through the GCMS, well, they're going to be almost identical. So it makes it really complicated from the research perspective of how do you identify these individual units and how do we classify these oligos as we biochemically can classify them and understand the function that they're playing. So there's still a lot of research going on, and I'll point out a variety of different areas of where there's some current research that's ongoing. A couple of notes. We have a few different enzymes that become very important with respect to our oligosaccharides, and the polysaccharides. So glycosyltransferases are going to be enzymes, you already know that, I know, with the ACEs, enzymes that transfer a monosaccharide or some sort of carbohydrate derivative. And if you remember back to when we looked at galactosemia and galactose being converted to glucose, we saw the role of the UDP, so the uridyl nucleotide involved in these transfer events. So that uridyl diphosphate that's linked to the ribose sugar. So you see these sugar nucleotides again playing a role. They all kind of play their different role, if you will. So we've looked at ATP and ADP and GTP and GDP, and here's UDP playing this role that it transfers these different monosaccharide units. So in this case, it's transferring an N-acetyl galactosamine and linking it then to a protein. So these transferases are important in that respect, that they transfer these carbohydrate units onto things that normally you wouldn't maybe expect to have a glyco portion. Keep in mind that proteins, but 50% of them, and enzymes too, but 50% of our enzymes contain some sort of carbohydrate moiety. So it is extremely common, and this is a huge area in biochemistry that we haven't even talked about yet, in which these enzymes have a portion that is a carbohydrate or have a carbohydrate linked. The way that occurs is by these glycosyltransferases. The group of enzymes that also does the hydrolysis of these glycosyl groups is also becomes important, although we don't talk about it too much, but keep in mind that there's enzymes that do this linkage and the transferring. There's also enzymes that will cleave off these different carbohydrates as they're formed. We'll be talking later about how these are linked, but right here you'll see where it's being transferred on is onto the side chain of a serine residue, and it's showing that below as well that we can link through serine. Serine is a common linker, again, providing the oxygen from its side chain to make these connections between the enzyme or the protein to the portion that is a carbohydrate. The area of research or biology that looks at the structure and function of these carbohydrate units is called glycobiology. And we also have a real trying to classify and understand these carbohydrates and the role they play, as well as in the cells or tissues that are specific to some of these processes. So looking at glycobiology just for a moment, here we're looking at a glycan group, and you can see overall our chemical structure being shown, and it's linked in this case to a protein through the N-terminal side chain of asparagine from that amide that is the side chain from asparagine. Asparagine and serine are both very common amino acids to link to carbohydrates. And then you see how we can shorthand by writing out these units similar to our amino acids, that we have these codes, and we're showing alpha or beta linkages in the glycosidic bonds between those different subunits, alpha, beta, that's what that's referring to. Or we can simply use those kind of cartoon shorthand colored shapes to show what carbohydrates are being attached and then which ones are being modified from their typical structure. So you don't see the glucose here, but we do see galactose, and then we see the modified glucose of glucosamine. So those are both being shown there. The four principles of glycobiology include glycan biochemistry, the biosynthesis of glycans, looking at the diversity of these different sugar units, and then understanding how they're being recognized and interacting in terms of a tissue or a cell. So looking at the figure in your book of 13.3 kind of shows the role of these different carbohydrates, or at least a few of them. We've looked at a little bit or thought about how glucose comes into a cell that uses transporters, because all of these carbohydrates and these monosaccharides individually are going to be extremely polar. They need to have a shuttle or a transporter protein to bring them in. So for glucose, that's going to be one of the GLUT transporters, GLUT. Comes into the cell, and then it can be broken down through glycolysis, or we can use it, maybe do some shuttling around or modifying of it to be used for formation of polysaccharides or the glycoconjugates or oligosaccharides. For the glycoconjugates that are made, some examples are shown here. We have this, I'm going to zoom in for a moment, cell surface glycoconjugate, in which we have this membrane-bound protein, and then it has a carbohydrate chain that hangs off into the extracellular matrix. On the other side of that, is a receptor. So this is a receptor that is able to bind to that ligand of the glycoconjugate, and it recognizes very specifically that carbohydrate that it's binding to. In this way, these carbohydrates are playing a very important role in these cell signaling and recognition events. So these carbohydrates being bound to a receptor, that receptor is able to have some sort of signaling with the other cell that is allowing it to interact. There are also, if you notice, the ER Golgi is being shown here. That endoplasmic reticulum is really important for the synthesis of these glycoconjugates that get sent to and incorporated into the lipid membrane and then have that extracellular carbohydrate moiety. Some of these are also playing an important role, perhaps within other portions of the cell, and some of them will stay kind of in that ER lumen to perform some sort of receptor signaling events, as well as protein-protein interactions. Overall, we have a few different types of oligosaccharides that we're going to look at. So the oligosaccharides, these are going to be from, 3 to 10, 3 to 20. We kind of have a little range. They're not looking at anything larger than 20. That might be branched or unbranched sugar residues. So some examples from plants are given here: stachyose, raffinose, rubascose, and they're all shown in the structure below. You'll see that raffinose has a sucrose unit that then is attached to a galactose. So all of these are going to be what we call these hetero oligosaccharides, in that they are not repeating units of the same individual monosaccharide. The rubascose has one more galactose than raffinose does. Rubascose has one more than stachyose. So all of these, if you notice, again, these glycosidic bonds become really important in the structure as well as the function for our oligosaccharides. If you notice, for each one of these, we are looking at a beta 1,4 glycosidic interaction. So for a lot of these, the fructose connecting to the glucose, keep in mind that one's going to be the alpha beta 1 to 2. Fructose is inverted and flipped, so its anomeric carbon is actually over here off that sucrose unit. But then if you look at the galactose linking to these glucoses, that is going to be, so writing over here, so carbon number 1 from our glucose is going to be there. And then this is going to be carbon 6 from galactose. That's going to be our anomeric carbon, or carbon number 1. So we'll see a variety of some of these 1,6, 1,4 from the sucrose. These are always alpha beta 1 to 2 linkages that start occurring. The galactose appear to be a beta 1,6 linkage over and over. Much of the time, we don't have the enzymes that break down. So if the enzymes are lacking to break these bonds, say if enzymes are lacking to break that glycosidic bond, this results in us not being able to break them down. This is going to then equal what we call dietary fiber, or your roughage from your vegetables, in which it's going to pass right through. If an animal doesn't have the enzymes to break it down, but in their gut they have bacteria that do allow that breakdown, the bacteria are going to digest them and then start to ferment on them, and that's going to create a lot of gas, which is very similar to humans who are unable to break down lactose but have bacteria in their stomachs that will, and it becomes very uncomfortable, that very uncomfortable feeling. So you can include, for example, animals in their feed. Some enzymes have been made that you can add to the feed, and then it starts breaking that down while they eat it to produce less gas and less uncomfortable feeling. The same thing for us. We call it vino, and it comes in a little white bottle that you can buy over the pharmacy counter. You know the commercial. Okay, so for humans, some of the interesting oligosaccharides can be, just as an example here, found in breast milk, and I've underlined a few kind of interesting notes, if you will. There are so many oligosaccharides that have been detected in breast milk, but only a few have been chemically characterized. And part of the reason, again, is as we have advances in mass spec, we're able to determine some of what these oligosaccharides are. But again, these individual carbohydrates have very extremely similar structures and extremely similar masses, and so it becomes difficult to really determine the correct size, and then therefore also attaching with that what the correct oligosaccharide is and the role it's playing. Some of the research that's come out on breast milk is really interesting, and maybe I kind of think that now after having kids myself, but recently I remember reading articles right after I had my second child, and she's only 5. So, so it's not been that long ago on some of these articles. A research team had found within breast milk a variety of oligosaccharides that can't be broken down by humans. And it was at first kind of a shock of why would these oligosaccharides be present if humans can't even break them down? After all, this is to provide nutrition for infants. From the consumer side of this, so even if you haven't had kids, everyone, has an interest in making the world a better place, or at least my students do. I tell myself that anyway. Or they like to make money. Or maybe both. You can do both. And with that, from the consumer side, becomes developing to meet nutritional needs of infants, the idea that you want to replicate as closely as possible for baby formula what is made by humans. And so because of that, the manufacturing companies that are formulating infant formula have kind of had a shock of, whoa, what's going on? There's these oligos that are present in breast milk that aren't actually being broken down by humans. What they're actually being used for is the gut bacteria within infants. So if you think about the advances that have been made, and especially biology probably hears about this more maybe than my chemistry students, about the advances in what we call our gut microbiome. So thinking about the bacteria that are present in a person's stomach that helps them digest their food, that helps them mount proper immune responses and have a variety of different cell signaling events from that side. A lot of these bacteria also help get rid of, pathogenic bacteria. And so these oligos that can't be broken down by humans that are present help feed those good bacteria. So let's call them good. And it provides, nutrients or food for these bacteria. These bacteria also help protect against pathogens and help us with our digestive system. They help infants and babies much the same way. And the gut microbiome and microbiology is really a huge area that has kind of exploded in its understanding and its research in kind of the current technology of where we're at with that knowledge. Then we'll also have these, so the good oligos. Then we have what we call, I'm going to call them fake, and put little quotes around them because they're not fake oligos. But these trick pathogenic bacteria into thinking that they are like the oligos shown in blue. And they bind, they try to feed on them, but they act as kind of like a suicide inhibitor where it will cause that bacteria to go through a cell death experience. And so it helps as part of our immune response. And this is also thought to happen within the guts of infants as they drink their mother's milk or, formula from a store that's trying to replicate as closely as possible their mother's milk as well. So, still a lot of research going on there and understanding overall these oligosaccharides that are present and what overall their function is, because not all of them are being used from a nutrition standpoint for infants. Some of these structural carbohydrates that have been well characterized, now we're looking at not just oligosaccharides, these are going to be polysaccharides that are going to be very, very large, huge repeating units that we're going to look at. The first is cellulose. And so cellulose is a homopolymer that we are repeating units of what we call cellobiose. So cellobiose is the disaccharide, and you'll notice the linkage here is going to be a beta 1,4. So we're linking from our anomeric carbon in a beta conformation from carbon 1 to carbon 4 of its neighboring unit. And this is going to be a continuing repeat. So cellulose overall is that huge structure. Cellobiose is just that disaccharide unit. Generally, as a general rule, what I tell my students, if you see beta 1,4 glycosidic bonds, this pretty much always equals a structural role, and I'll show you why down below here in a minute. Hemicellulose and pectin are also polymers that are mentioned in your book. We're not going to take a huge look at them there. You can, if you will. Hemicellulose is branched, unlike cellulose that is not. So cellulose are these repeating huge long strands. What starts to happen is you'll have the one strand interacting with the other, and it creates, because of these beta 1,4 glycosidic bonds, rigid sheets. And these rigid sheets are awesome in their function of the cell wall, where they're providing structure and protection for the plants. Tree bark is an example of cellulose. Almost the vast majority, at least, of tree bark is cellulose, and you'll see this in all of the plants. As they form these sheets, there's hydrogen bonding that happens between these from one strand to another. And with all of this hydrogen bonding that's occurring between these strands, they largely become insoluble in water. Okay, so the hydrogen bonding promotes low solubility. Why? Because they're hydrogen bonding with each other and not with aqueous. Low, I'm thinking and talking at the same time. It's always a problem. Promotes low solubility because they're not interacting or hydrogen bonding with aqueous solution. They're hydrogen bonding to each other. And we'll see very similar things even with our other alpha linkages, and ask my students who have had to make some of these polysaccharides, especially like starch, in water, and you can make a low concentration and it just does not want to dissolve. So you almost make like a slurry because of that hydrogen bonding to each other. You can see in this cryo-EM of a plant cell wall, that cellulose structure here. It's pretty cool looking. Another structural carbohydrate is going to be chitin. This is going to be linear branches, or a strand. I say branch, I mean like a linear repeating strand of units. These are going to be the N-acetyl galactosamine that we have, that N-acetyl group that's linking, and it kind of decorates each side. So in this case, these beta-1,4 glycosidic bonds, again, are playing a structural role. So if you see beta-1,4, they're very rigid. In this case, they are being used as the exoskeleton for insects and crustaceans. So if you step on a bug and hear that crunch, that's carbohydrate all the way. The mushrooms that are shown, their cell walls also have chitin in them. Okay, quick note on our cellulose and our chitin. So cellulose, you require enzymes to break these glycosidic bonds and to do that hydrolysis reaction. Just as a reminder, we do not have the enzyme that breaks this down. So the enzyme that breaks that bond is cellulase for cellulose and chitinase for chitin. And that's going to then perform the hydrolysis of that beta-1,4 linkage. And so for some animals, they are able to do that, or they have the bacteria, of course, that are able to break those down. Cellulose for us is going to be what we call a low-carb option. Is it low in carbohydrates? No. It is not going to add to your carb count because we can't break it down. So chitin, the enzyme that is able to do this hydrolysis, will be chitinase, cleverly named. Okay, so that takes care of a couple of the important polysaccharides. We'll look at starch and glycogen in a moment, and later on we'll look at the end of this lecture, we'll look at the role of bacterial cell walls. The next one I want to look at, though, are the glycosaminoglycans, so GAGs. These are going to be those hexosamines that are repeating, generally 20 to 50 disaccharide units. So our proteoglycans, chondroitin sulfate, heparin sulfate, keratin sulfate, all play roles here. What you'll notice is they all have the sulfonic acid that's attached through the oxygen to the carbohydrate unit. So chondroitin, keratin, heparin sulfate has that sulfonic acid group that's attached. Very polar, lots of electronegativity around that group. The function for these sulfates, so the role of the keratin sulfate in the human cornea is discussed in your book. Very interesting. It makes kind of these, how do I say, like a sheet over your cornea, and allows you to properly see. Whereas if the sulfonation is incomplete, the cornea is not protected in a way that, obviously you can see that film almost that looks like it's over the eye, and you have degeneration of your eyesight that results from that. So very important functional role there. The next one I want to talk about is starch and glycogen. These are homopolymers of glucose. We have glucose units that repeat and repeat and repeat. Both of these are going to play a role in our energy. So let's turn my brightness back up here. So starch is composed of two different types of polysaccharides, amylose and amylopectin. Amylose is going to be a linear alpha 1,4 glucose linkages, whereas amylopectin is going to be a branched molecule that branches off of the 6th carbon in an alpha 1,6 linkage. This is going to be an energy molecule, and its energy storage here, storage for plants, whereas glycogen, very similar to starch, glycogen is going to have also linear alpha, let's fix that, alpha 1,4 and then alpha 1,6 branching points. It just branches more than what the amylopectin does. So on average, amylopectin branches around every 20 units. Glycogen has the branching of around every 10 units. But keep in mind that's a range. It's like 8 to 12. And it's like, I don't know, 18 to 22 or something. So it does vary. Overall, glycogen is going to be the energy storage for animals. So plants do not have glycogen, they have starch. Animals don't have starch, they have glycogen. The amylose structure is going to result in this spiral, 3D spiral molecule because of the alpha 1,4 linkages that cause it to kind of spiral around. And you'll also see a lot of hydrogen bonding, not to each other so much as what we saw with cellulose, but it will still, there's still a lot of hydrogen bonding within that network. They don't quite align as nicely as what we saw with the cellulose into those rigid sheets. The amylopectin and glycogen structure you can see here below, so the alpha 1,4 linkage, when we generally see alpha 1,4, that tells you it's an energy molecule. The beta 1,4, structure. And then these alpha 1,6 points that branch off. The branching for glycogen and starch is shown here. So what this looks like is like a top view looking down. Starch is shown above, glycogen is shown below. And what you'll see from glycogen, all these branching points give this a greater surface area. Or if you look at this ball thing here, koosh ball, I didn't know that's what it's called. My daughter had one. She called it a sticky ball. My sticky. The glycogen is going to have more of these like legs or arms, whatever you want to call those, I don't know, off of it. Whereas the starch is going to be like this bigger one that has less of those. The greater surface area in glycogen is going to allow for more energy to be released in a shorter amount of time. Why is that important? You know, and especially the role that it's playing in the animals versus plants. Well, plants don't get up and run around like animals do, and therefore animals need to be able to tap into that energy source through the hydrolysis of those individual glucose units, and this allows that greater surface area a faster energy response for animals than what plants require. So 13.2 starts to look then at the glycoconjugates. This is going to include the glycoproteins, the proteoglycans, and could also include the glycolipids, although we'll look at those perhaps later when we look at lipids, but they are kind of referred to or mentioned. Glycoproteins, just that definition again, the bulk of that macromolecule is going to be protein, so we place the emphasis on the last portion of the glycoconjugate. So proteins being more protein or more amino acids, proteoglycans, they're going to have more carbohydrates. Okay, so the functions of the proteoglycans are going to be multifold. They are going to help regulate signal transduction and cell signaling events, allow for cell migration to occur. So there's really two classes of these, cell surface and extracellular matrix, just based on the location that they're found. So these proteoglycans are largely made up of carbohydrates that are then linked to maybe a peptide or a small protein. what you'll notice is these large repeating units, and especially like what we looked at with the kerosene sulfate, the chondroitin sulfate, these N-acetyl glucosamines or galactosamines that are going to be observed to occur, they're going to be found in the extracellular matrix, whereas the cell surface proteoglycans, nicotine, as an example, are shown where they are tethered to either a transmembrane protein in the membrane, or they're linked or anchored to a lipid. So this is going to be one of our glycolipids as an example that's attached or anchored into the membrane by linking directly to the lipid, whereas the heparin sulfate for the other ones has our core protein, and that protein is going to be found at that transmembrane domain and be anchored into the membrane by those domains. The carbohydrate moiety can link to proteins or enzymes in two manners, an N-linked or an O-linked oligosaccharide. And we call them that based on what amino acid is going to be providing the linkage from their side chain. So asparagine, as we noted above, links through the nitrogen, its terminal nitrogen from that amide linkage or its functional group. And that's going to give rise to our N-linked proteoglycans or glycoproteins, whereas the oxygen from the side chain of serine is going to be an example of one of our O-linked. The other example for our O-linked that you won't be surprised by is going to be threonine. So our N-linked is going to be asparagine, the O-linked is going to be serine or threonine. Tyrosine also has the potential, but because of its size, generally doesn't link to carbohydrates, but there are some examples where tyrosine does much of the same way that these amino acid side chains provide the oxygen to link to phosphoryl groups. They provide an oxygen that can easily be linked to the anomeric carbon, and so that's something that should be noted, that these link, both of them, through the asparagine or the serine threonine. This links to the anomeric carbon. The glycoconjugates, how they function in cell signaling and immunity, we're going to take a look at a few examples of how these are important and why. They can have what we call intrinsic or extrinsic interactions. The intrinsic interaction and recognition events happen between identical, proteins or cells from the host. So this is within the one organism, where here you have a host cell that has this glycoconjugate that maybe it's a carbohydrate coming off of it that's going to link to a receptor molecule that we would say intracellular adhesion, or it's within the same cell. Can also have intercellular adhesion when there's two host cells that are being linked, as was shown above, between a ligand and a receptor. These receptor molecules are called lectins that are recognizing and binding to the carbohydrate portion. Extrinsic recognition events are going to be between two cells from different organisms. Here you have adhesion as an example between a pathogenic and like a lectin from a pathogen and a host cell. So for example, a pathogen cell that has carbohydrates coming off of it that can be recognized by lectins from the host cell, or vice versa, where the host cell's carbohydrates can be recognized by a carbohydrate binding lectin protein. So these lectins, the definition is going to be these are carbohydrate binding proteins. Often these are going to be the receptors that specifically recognize a carbohydrate portion from a protein or from a lipid that's hanging off in that extracellular matrix, and then it's going to bind as a ligand to that carbohydrate portion. So shown below is an E. coli cell, and it has these lectins that are shown in green. So the structure shown here of that lectin protein that's going to bind to the carbohydrates from the host cell. Bacteria can do this. There's also a virus particle that's being shown here through its carbohydrate portion through lectins that are bound to the external virus particle that can recognize the carbohydrates from the host cell. Or you have the host cell able to recognize the host cell. This is going to be, instead of an extrinsic event, this is one of those examples of those intrinsic binding events. The other, another function or, I guess, importance of these oligo or glycoconjugates, some of them oligosaccharides or polysaccharides, becomes in with our ABO blood typing or ABO blood groups. So here, about 75% of this contains glycoproteins and another 25% of glycolipids. And these are going to then be examples of these glycoconjugate molecules. So shown on the left is our O antigen, and you can see our sugar code with those different symbols. And we have 4 oligos that are drawn out for you, and that's going to be the O-type blood. The O-type blood lacks the A or B GT enzyme, so they don't have the GTA enzyme or the GTB enzyme. These GTA and GTB enzymes are, in this case, a type of transferase that are going to transfer a carbohydrate portion onto that O antigen and make a different antigen. So the A-type blood has the GTA transferases that are active. The B-type blood has the GTB transferases that are active. AB-type blood is going to have the activity of both the GTA and the GTB transferases. And so each of those, the B-type and the A-type antigens, are being made. So the O antigens does not have either the A or B transferases. Keep in mind that this is different than the Rh factor and the antigen that gives rise to your positive or negative of your blood type. This is just type A, type B, type AB, or type O based on the activity of these transferases. So at some point in recent evolutionary history, there's been a divergence of what of these transferases are active, and not everyone has the same ones that are active, and that's what gives rise to our different types of blood. As you are all probably very familiar, because if you're in this class, that type A, type B, AB, and O cannot universally be given to another person. So a donor has to have a match with the receiver, if you will, of the blood for a blood transfusion. And, it wasn't all that long ago. I remember as a kid hearing about people dying from mistakes being made at a blood bank or something, or at the hospital where things were mixed up and someone died because somebody was given, a transfusion was done with somebody who was not an acceptor. And I mean, it wasn't like it was happening then, but it wasn't that long. I guess I'm old. So I, really hope that these things don't still happen, because if someone has an antigen or antibodies being made for the antigen that's being given to them, it results in lysis of the red blood cells, and it is lethal, and therefore the receiver of that blood is going to die. So from that standpoint, the way that the reason for this is here in these glycosyl transferases. So for example, someone that has type A blood, and if you're looking down this column of type A blood, they have the A antigen that's made, they have the transferase, the A transferase that's active, and therefore they're going to have antibodies be made against the B antigen within their plasma. The type B blood, on the other hand, so the next column as you look down, have antigens that are going to be made for, or the B antigen that decorates the outside of those red blood cells, but they're going to have antibodies for the A antigen. The type AB has both transferases, both antigens that are present on the outside of their red blood cells. They are not going to have in their plasma anti-A or anti-B antibodies that are made. So they are going to be what we call a universal acceptor. So the compatible donors for type AB, because they don't have antibodies made against A or B, they can accept from A, B, AB, and O. So they become the universal acceptor for red blood cell donors. Keep in mind, the plasma is different. Type O blood does not contain either A or B transferase enzymes, so they contain neither A or B antigens decorating that red blood cell. But because of that, within the plasma, they have antibodies for both A and B. So because of that, here, the red blood cell donor can only be for O, but they are able to have plasma donors from O, A, B, or AB. If you'll notice, because the type O doesn't have those antigens that decorate the outside and don't have from their red blood cells, they're going to be able to donate to type A, type B, or type AB, or type O. So these type O becomes the universal donor. Because of that, there is a high demand for type O blood, and it's a very common and I don't know, popular blood type. So what happens is hospitals tend to use O a lot, and therefore there's a shortage that results from the type O blood. So if you are type O, I strongly, whether you have positive or negative antigens or the Rh factor, if you're positive or negative for that, it doesn't matter. If you're type O blood, donate. Hospitals are constantly, and especially recently, running short, and I know because they call me every single time I'm able to donate. So donate blood, save lives from that. But it becomes really interesting of how A, B, and O type blood typing is, and it's because of these carbohydrates that decorate the outside. Another area of research, understanding antibiotic development and how antibiotics work. Arguably the most important and significant medical breakthrough in modern medicine has been the development of antibiotics. And the way that this works, so bacterial cell walls are composed of these repeating units of peptidoglycan, and it's N-acetylglucosamine and N-acetylmuramic acid, those two carbohydrates, that disaccharide, that repeats over and over and over. The gram-positive versus gram-negative, for my biology students that have taken micro, you're likely familiar with the differences because you have to do gram staining in micro. The gram-positive has that peptidoglycan cell wall, and it's going to be nice and thick, whereas the gram-negative have a peptidoglycan cell wall, but it's going to be much thinner than the gram-positive. The gram-negative have an outer membrane that's shown in this green on the outside of that purple peptidoglycan cell wall, and therefore they have a difference in that cell structure of their outer cell walls. Shown in this cartoon is that kind of peptidoglycan cell wall layer and that peptide chain that's linked then to those two repeating units. So the way that penicillin, the first antibiotic that was developed, works is it's a suicide inhibitor of a transpeptidase that's important for the synthesis of bacterial cell walls. So shown below here is this beta-lactam ring that is penicillin. So it's a structural analog of the natural transpeptidase substrate. Therefore, it inhibits that transpeptidase that's important in making the cell wall of bacteria. So as bacteria mount an offense to a host cell or a host organism, they'll start trying to first build how many bacteria are present and then, start attacking, if you will. And the way that bacteria are able to communicate with each other, it's quite interesting. A lot of carbohydrates that are also oligos that are important in that. But as the bacteria start to try to replicate and divide and make more of themselves, they're unable to build their cell wall, and therefore they die. And that's how penicillin works. And it's because of that lactam ring that the natural substrate doesn't have, and now it's not able to do the chemistry to make its cell wall. So the latest epidemic for our antibiotics is that these bacteria are able to develop or to, make an enzyme that's going to overcome what that suicide inhibitor, that antibiotic, has done. So a classic case is our MRSA, our methicillin-resistant Staph aureus. And in this case, this bacteria, the Staph aureus, is able to have an enzyme that's going to overcome penicillin or methicillin. So methicillin, shown below, and you can compare the molecular structures to penicillin, shown above, very, very similar. It's just a slight modification of penicillin is our methicillin. And it's also going to cause suicide inhibition of a transpeptidase. And what has resulted, so if you look at penicillin-sensitive bacteria, they have that transpeptidase that becomes inactive, and then the cells die, and yay, people are happy. But when they have a beta-lactamase that can break that beta-lactamase ring, or the beta-lactam ring, then they can open that up and therefore overcome that inhibition. The bacteria can make their cell walls and they live, and then, well, we can get sick and die. And so then we're unhappy. So the happy or sad faces are for humans. The methicillin-sensitive bacteria, same idea. That when it's sensitive, people live, and when the enzymes are made, those transpeptidases that can overcome that inhibition, then people die. And so there's been a large driving force for the R&D of new antibiotics in which organisms aren't going to be able to overcome, or that we just simply have something else to tap into as an option with our antibiotics. Quick note on glycoproteins and a little bit. So we looked at a lot of these different glycoconjugates and their functions and what the role that they play. Glycoproteins, there's so many of our proteins that have a carbohydrate moiety of some sort. And these carbohydrates are going to be attached via their anomeric carbons, and with that resulting in a lot of cell signaling events, protein-protein interactions. They are important in the immune response. It goes on and on and on. Bacteria, for the most part, have very few of their proteins that are glycosylated. And because of this, we are able to identify the pathogen and mount an immune response effectively against it. And so, effective immune response, people live and we're happy. Viral proteins, on the other hand, are heavily glycosylated, meaning that they have a lot of these carbohydrates that are attached to them. Because of this, that they are able to kind of, especially for eukaryotes, be able to trick the host cells, and the host cells don't recognize that there's a pathogen there, or some sort of other component that is going to overtake that host cell and then use its proteins and enzymes to do what it wants to do, and that's to make more virus particles. And so because they're heavily glycosylated, they trick the host cells into not recognizing that there's some sort of pathogen that's there. I included this table just to give you an example of some of the glycoproteins. Some of these, for example, the immunoglobulins that we talked about previously have a carbohydrate portion. There's some different hormones or membrane proteins that are also going to be these glycoproteins. So we looked at lectins in this lecture a little bit and how they act as a receptor, and by specifically recognizing and binding to a very specific carbohydrate. And the way that they bind to those, again, those interactions are going to be hydrogen bonding, hydrophobic effects, van der Waals interactions, etc. So your O-linked versus your N-linked, again, for this becomes a big takeaway, that the O-linked are your serine and threonine, and your N-linked is the asparagine. These are the three most important amino acids in terms of our glycoconjugates and the way that they're made. These glycoproteins are going to have a wide variety of functions, many of what we've already talked about. So, hope that's helpful. Hope you're enjoying this lecture, and I'll see you in class.